principled and kind of ready to go. The overarching gist of the talk is going to be looking at the different ways we measure interoception and brain-body interaction, and trying to ask if we can develop better measures that have better face validity, construct validity, test-retest reliability, the kinds of things that are important both for the basic research of understanding brain-body interaction and the applications in psychiatry, and maybe someday something like interoceptive computational psychiatry. So I think the audience is kind of a mix of different backgrounds. So we start with a very general introduction of what is interoception. At the most general level, we can think about the interaction between the body's internal visceral organs, the heart, the lungs, the stomach, more widespread systems such as the endocrine system and other metabolic systems. And the way that these organs in their activity of homeostasis keep us alive and keep us processing air and pumping blood and digesting food, where we can generally think about all of these different visceral rhythms as kind of a clock-like signal that is involved in the sustenance of homeostasis. So I think the first most general distinction that's useful to make is between something like implicit interoception or brain-body interaction, which might be phenomena like if you see an exteroceptive or an external stimulus differently, if it's presented when your heart contracts or when your heart relaxes, or if breathing in time with a stimulus alters the way that you learn about it or encode it. Those are kind of things that you're not necessarily consciously aware of. They might relate to having a higher overall conscious interoceptive awareness, but they're kind of at this implicit or subpersonal level of synchrony between the brain and the body. And then we can think about more explicit forms of interoception, which is the way that you sense, perceive, become metacognitively aware of, and ultimately control those visceral sensory and visceromotor signals. So that's things like, as I start talking, I feel that rush of adrenaline, and I can notice that my heart is racing, or I can feel that my tummy is rumbling or something like that, and the ability to really monitor that ongoing bodily activity. So a very useful psychological model that was developed by Sarah Garfinkel and Hugo Critchley to kind of divide up this realm of explicit interoception is basically a three-part model where you can think about interoceptive accuracy being the objective sensitivity you have to bodily states, things like can you detect a heartbeat or not, or can you detect resistance on your respiration or not. What they call interoceptive sensibility, which is more the subjective or self-report level, like if I ask you a question, do you typically notice when your heart is racing? And what they call interoceptive awareness, which is really a kind of just metacognitive awareness, or if you're from a clinical background, you might know the phrase insight. This is, not how objectively good at detecting a cardiac signal or respiratory signal are you, but how well do you know if you're good or not? It's kind of statistical correspondence between things like subjective confidence or awareness ratings and objective ground truth accuracy. So these things have been, there's been a lot of studies on these three different levels and different ways to interrelate them. We'll kind of delve down into them specifically in the cardiac domain later, but it just serves to have a nice background between these different domains of interoception. But I'd like to skip to why do we care about interoception? Oh, is this little sharing thing blocked? Sorry, guys, I'll just move that. Why do we actually care about interoception from a kind of, community or greater good perspective? And I think there's a really nice kind of summary statement from the field coming out of a summit at the National Institute of Mental Health with many, many authors led by Sabib Khalsa, where they made this kind of review of the overarching transdiagnostic nature of bodily symptoms and interoceptive symptoms. The idea being that when you look across panic disorder, depression, eating disorders, generalized anxiety, autism spectrum, and so on, there's a lot of sort of embodied phenomenology that have to do with different visceral systems or gastric systems and so on, which have traditionally been treated as kind of almost epiphenomena of the mental disorders themselves, something that is, not a part of the causal machinery of what gives rise to the mental illness, but just a kind of side symptom. And the review is making the case that we need to start to think about these underlying symptoms as part of the what actually could give rise to mental illness. And that's something that I think we see increasingly coming to the forefront in psychiatric research. I was at the Royal College of Psychiatrists in London about 2 years ago, and it was kind of remarkable to see so many keynote lectures and talks about, the way that interoceptive and bodily factors might act as either increased risk factors or kind of mechanisms in a runaway positive feedback loop. Here I put on the screen just one very general example of that. This is coming from the literature on depression and obesity, where we know that the odds ratio for developing depression if you have obesity is, I think it's something like 1.6, and vice versa for if you have depression to become obese. So we know there's a strong causal link between depression and obesity. And what you're seeing in that field is an increasing dependence on sort of multi-risk models, where in addition to genetic and lifestyle, environmental, and sociocultural factors, you have things like inflammation and disordered interoception that are really acting as a brake or a accelerant in this ongoing loop. And then when we look into the neural models that are proposed for these things, we see our usual suspects of kind of very closely related to interoceptive brain regions, such as the anterior cingulate, the orbital frontal cortex, the hypothalamus, and the insula. So this is just one kind of an example of where there's an increased interest in understanding how we encode and learn from and perceive bodily signals so that we can have a better understanding of mental health. We can zoom in further. So something that my lab has been really engaged in over the past few years is trying to develop conceptual and computational models that explain how different exteroceptive and interoceptive signals are combined in the brain to give rise to decision-making and explicit interoception. For example, in this review, we were looking at some recently published studies in rodents using optogenetics and manipulations of things like food and satiety signals to propose a kind of expansion of previous work, which proposed that there's an important role for interoceptive prediction errors in shaping emotion and different affective states, where here we were zooming into the insular cortex specifically to look at the literature and propose that you have a kind of a gradient or a sieve-like function where, because of the specialized cytoarchitectonic and connectivity structure of the insula, it's kind of an ideal structure to integrate exteroceptive prediction signals with interoceptive prediction signals, and then to read out a translation of confidence or confidence prediction error, which is a kind of a second order expectation on the reliability of those two systems. So this is something you can have a look at for kind of trying to get down into the nitty gritty of those signals. And this kind of idea of balancing between interoceptive evidence and exteroceptive evidence to come up with some metacognitive self-representation is something that we've expanded on more generally in our so-called interoceptive self-inference model, which here it's a similar kind of idea. We take the standard story from reinforcement learning and decision-making literatures about how we learn and update our models of the world, where, for example, in the exteroceptive channel, you've got sensory prediction errors that are conditioned by their precision, their gain, their confidence, as you like, where at some higher order level, you've got a readout of those confidence signals, and then at some metacognitive level, you've got a prediction of expected uncertainty. So a kind of top-down model, not just of prediction errors or cross-modal prediction errors, but of the kind of regularities of prediction errors. And a part of what we've been trying to hypothesize is that at this metacognitive level, it makes good sense for the brain to be monitoring interoception in the same way we know that it monitors exteroceptive precision, to try to come up with some kind of overall estimate of expected volatility or expected uncertainty. And the kind of cartoon of this model is that, if you assume a sort of baseline, stable, syncopated breathing state or a reliable heart rate variability state, and then, something really frightens you or something unexpected happens that causes you to hyperventilate, or you have organ failure unexpectedly, this is going to ultimately result in a spike in the noise that's encoded in your different exteroceptive sensory channels, and your brain can exploit that interoceptive information to basically alter the salience or how much it cares about those changes. So this is something we're working on writing down into a computational model and testing empirically, with the idea being that if we could hold certainty on one channel constant and manipulate it on the other, we could start to actually disentangle how these systems interact with one another. To give a slightly more concrete example of what we mean here, or I should say a toy example, really, you can think of something like the way visual information enters the retina through the optic nerve, where at each heartbeat you have a kind of systematic compression of the optic nerve, resulting in a kind of cardiac locked sensory attenuation, or attenuation of the ascending sensory inputs. We were interested in this kind of general phenomena as an example where you might have a coupling between the sensory noise level of your exteroceptive systems and your interoceptive processes. And we tried to build a computational model of this process. I'm not going to have time to go really deep into the details here, but we can always talk about it later. The essential idea here is we built using the Markov decision processing framework developed by Carl Friston and colleagues, a simple little simulated agent where the agent has its own cardiac states that it oscillates through in a reliable manner. So it goes through a certain sequence of systolic and diastolic heartbeat states. And the agent is confronted with exteroceptive events that have been given a kind of inherent valence in terms of prior preferences about which items it should sort of approach or avoid. And the great thing about having a model like this written down is that we can go beyond kind of conceptual theorizing about the link between interoceptive or exteroceptive prediction errors and actually go into the agent and lesion the precision or the confidence of signals arising from one channel or another, and then try to simulate how those lesions will affect behavior and physiology. So that's something that we did with this model. I should say, crucially, the relationship to the cardiac, the depression of the optic nerve I was talking about is that in our model, we were trying to get a grasp on these frequently reported effects where you see, for example, that fear processing or perceptual processing is enhanced during or suppressed during certain phases of the cardiac cycle. So we, in addition to this agent having these two different sensory channels, we equipped it with a kind of periodic sensory attenuation. So each time the agent's heart beats in the final phase of the cardiac cycle, we attenuate the ascending sensory information. And the goal there was just to see if you take a really toy, biophysically unrealistic model and make it so there's just this minimum interaction between interoception and exteroception, what type of phenomena can you reproduce? So when we do that and we simulate a kind of experiment where the agent encounters, say, 15 trials of something that's not startling or a kind of a baseline stimulus, and then on the 15th trial, we present an unexpected threat stimulus, a spider, if you will, we can see that the agent's prior beliefs of encountering a spider on the next trial, they kind of spike after that spider is presented, as you would expected. But perhaps more interestingly, when we look at the healthy agent in red versus the agent where we've gone in and lesioned all the interoceptive connections in blue, we can see a few different things that emerge. So one is that in the baseline period, the lesioned agent is already having a kind of heightened expectation of encountering these threat stimuli. And in the evoked period following that, it actually has an attenuated belief update. So to put it another way, the model reproduces something like a kind of startle reflex, which basically just demonstrates that when you have a very simple model which hypothesize a cyclic link between interoception and exteroception, you can produce experimentally plausible behavior. We also use this agent to create synthetic ECG traces just by running it for a very long period and treating the synthetic heartbeats as RR intervals. And the purpose here was, again, not to provide a generative model that you could fit to your current cardiac data, but just as a proof of principle that in a simple agent like this, where you link up these systems in this way, it's actually possible to produce synthetic HRV traces that are idiosyncratic or unique to different clinically interesting cases. So the case where you have a healthy agent where nothing has been lesioned, or an agent that has a really hyper-precise visceral system, meaning the sensory signals in the visceral system are overly strong or salient, versus an agent with a hyperarousal prior. So that's something that we thought was pretty exciting from a kind of toolkit perspective, because it opens up the possibility that we could begin to move towards something like a computational psychiatry of the embodied mind, meaning if we want to better understand the nosologies and the distinctions, the classifications of different mental illnesses, and we want to do that in a way that respects the fact that certain sub-classifications could easily involve a very similar central nervous system, but a very different peripheral or autonomic or even visceral organ system, you want a kind of computational approach that can do that phenotyping, meaning that can make mechanistic predictions about the way the brain and body interact so that you can ultimately get a better understanding of the mechanisms and hopefully start to do a more targeted treatment and diagnostic approach. Now that sounds really, really good, and it would be really great if we could do that right now, right? If we could start measuring people's brain-body interaction and get really concrete understandings and interventions from that. The problem is that the measures we have right now to quantify interoception, meaning to quantify the kind of explicit ability to detect these signals, are very, very poor, or at least they've been the subject of a lot of intense controversy over the previous 30 years, basically. I'm not going to go into depth into all those criticisms, but if you're interested in that, I really strongly recommend starting with this review paper by Brenner and Ring, towards the psychophysics of interoceptive processes. That's a paper that our lab has taken as almost a mission statement. And you can just see this smattering of papers here, a lot of arguing over the different techniques. I realized I didn't say something, I'm sorry. So obviously this model here is more about implicit interoception. But what I mean to say about these models in general is they underline that a computational approach to phenotyping interoception, it requires you to have tasks that allow you to start to causally perturb and dissociate these different types of things, right? So you need to be able to have a level of specificity in your measurement that can allow you to dissociate things like autonomic prediction from autonomic uncertainty, or even better, cardiac uncertainty from respiratory uncertainty. So I think it's kind of a clarion call to basically have measures that are amenable to the types of modeling and the types of manipulations we see in other mainstream forms of decision-making and perceptual decision-making in the interoceptive domain. So that is basically, oh, and then I think the last thing to say is, you know, the Olivier Desmet and his colleagues, they're very useful when you give a talk like this, because every time I think I need a slide that says that the way we're doing it isn't working, it's like they have another preprint showing exactly that. I just briefly touch on this preprint they just came out with. So you've got the construct validity issue of things like the heartbeat counting and the heartbeat discrimination task that we'll get into it a little bit more in a second. And then you've got the kind of clinical validity or the relationship to interoception. You know, one of the things that I start this talk with, and I think every interoception paper starts with, is some preamble about William James talking about how important interoception is for emotion, and vis-a-vis must therefore be important for affective disorders like depression and anxiety. But at least when we look at measures such as the heartbeat counting task, which have been criticized for a variety of different reasons, in this recent, like literally this month, meta-analysis from Olivier and colleagues, they basically find either a very, very tiny correlation between, for example, heartbeat counting behavior and a variety of anxiety subscales, or absolutely no correlation with depression. So if we want to get towards something like a computational psychiatry of interoception, we've got to start to face this measurement crisis and start to develop measures that are specific, are valid, and are reliable. And I'm going to try to show you some of our efforts to do that, although I'm almost 24 minutes in, so I'm way behind time. I think the second time you give a talk is often the worst, so I'm sorry for that for people in the crowd, because this is a little less smooth than the first one, but it's okay. I think you're with me. So in summary, I think that computational modeling of interoception can, in principle, help us to better understand how the mind and body go together. You know, what I said in my grant interview for my Lundbeck Fellowship was, you know, you hear all the cliches about the complexity of the brain. Well, if we want to actually understand the mechanisms that integrate the brain and body, that's like an nth order expansion on the complexity itself, right? We need a reductionistic approach that can start to pull apart those pieces in a reliable way if we're ever going to get there. So we're going to look at a few different ways that we've been trying to do that. I'm going to zoom through this because it ended up really bogged down last time. We're going to start with just good old-fashioned psychology and subjective measures. I think we should, in the rush towards the psychophysics of interoception, we shouldn't neglect just asking people what they're feeling. So this is what is often called interoceptive sensibility, which is basically measured with a variety of different self-report inventories, things like the Porges Bodily State Questionnaire or the Multi-Dimensional Interoceptive Assessment, the MIES. There's actually about 15 different ones of them. Luckily, there's another preprint from Olivier and colleagues that makes it so I don't have to go into a lot of detail here. They actually just did a large-scale 1,000-in meta-analysis and construct validity study on these self-report questionnaires. And the kind of damning statement there was what you see here is a network plot. And basically what you can kind of eyeball right away, each of the colors is a different interoception scale, all of which are purported to measure the same underlying construct, something like how you feel about your body. And in fact, they recover just as many they recover just as many factors as there are questionnaires. So as they say in this highlighted statement, it's already looking kind of bad if you think that these 15 different measures or so are actually indexing the same underlying construct. And to be honest, we weren't very surprised when we saw this, because when we started working with these scales, I think I was still physically at Cambridge Psychiatry then, and our clinical colleagues were like, what are these scales actually measuring? They're asking questions like, how do you feel about your body? They're asking sensory questions, cognitive questions, metacognitive questions, like, I typically notice when my body is doing this or that. They bring in things about pain and discomfort and anxiety. One clinician said, this looks like it's half of an anxiety scale to me. And I think that kind of stood out to me, is that a lot of these scales, rather than sort of focusing just on what you're feeling in your body right now, are kind of expanding to many different constructs. So we wanted to develop something based in an experience sampling method, kind of borrowing from the mind wandering or self-generated thought literature, where we would try to get rid of a lot of that baggage and just have people rate via beeper method a vast scale along 12 different dimensions, or 12 different items of basic interoception. Things like feelings from your throat and your nasal passage, your heartbeat, your breath or respiration, your stomach digestion, your bladder and need to defecate, more what we call malaise symptoms like feeling tired or fatigue, feeling of headache, feeling of temperature, feeling of pain or aches. I'm going to zoom through this part. We did this as part of a COVID-19 experience study during March of 2020. For now, I'm just going to show you the data from time one, where we have about 600 participants. And I'm going to zoom over a lot of the different outcome measures because it's a little bit too involved for this talk. We did a series of factor analytic steps, including exploratory and confirmatory factor analysis. That will all be in the preprint soon, but the short story is, according to Roger Kiviet, it was one of the best fitting factor structures he'd seen for a first-time factor analysis, because he was basically tutoring me in it. Long story short, we get a pretty nice factor structure recovered from this data, which loads on kind of sensible modalities of interoception. Cardiorespiratory are both closely related. Gastrointestinal, including feelings of the skin, are all closely related. These kind of malaise factors cluster together, and there's an obvious airway factor. We use that to then examine, after confirming that there was structural invariance up to the level of partial invariance on the scale items. We examined whether having a prior diagnosis of a psychiatric disorder, such as anxiety or depression, was associated with elevations on these subjective feelings of the body. This is literally just in the past 24 hours, how much have you felt sensations from that body system? And what we saw was that there were substantial increases in the cardiorespiratory factor and the malaise factor. If we separate those by depression and anxiety, I should say that there's about 200 patients in total in this sample. We see that the depression is a little bit more specific to cardiorespiratory and malaise, whereas anxiety seems to be associated with general increases in subjective distress. In multidimensional interoceptive feelings. So it seems like in the context of a pandemic, where you are being told that your body might kill you, attending a lot to the bodily sensations is associated with having some history of mental illness. If we look further into that and we separate by persons who at time 1 said they had a dry cough symptom, we again see very robust increases across all of those multidimensional domains. So it kind of makes sense. If you feel like you might have COVID, you pay more attention to your body. And then we look at people's self-rated feeling of isolation distress. So if they answer the question, how much is self-isolation disturbing your mental health? We can see that the psychiatric patients are obviously robustly elevated in that variable, and that correlates strongly with the degree of interoceptive feeling in each domain. So the more you say that social isolation is hurting your mental well-being, the more you also report feeling sensations from most of the modalities. Actually, in the partial regression plots, you can see that the cardiorespiratory and the malaise are associated with an increase, are correlated with an increase in impact on mental health, whereas the airway and gastric factor seem to be negatively correlated. Although I wouldn't put that much weight on the partial correlations. So I know that was zooming fast, but I want to get to the psychophysics part. And I should have listened to my wife, who said to take this part out of the talk because it is a detour. I'm sorry, but it's so interesting data. The summary is that people often ask me if I think that psychiatric illness has anything to do with interoception at all. And I would say subjective interoception, definitely. Maybe not in the direction we'd hope. Maybe it's just about being in a pandemic that a lot of subjective attention to the body is distressing. But to me, this is at least, I don't want to say comforting, but it's some face evidence that just asking people about their bodily sensations does relate to psychiatric symptoms and distress. But, you know, herein lies the problem with self-report, right? We have no idea, is this like a kind of functional disorder or hypochondriasis? Is this just allocating a lot of top-down attention? Is this actually having a boost in bottom-up bodily signaling that is being correctly recognized by the perceptual metacognitive systems? We don't know. To know that, we need psychophysical techniques that can dissociate the sensitivity of different objective decisions about the body versus their bias. And that's what we're going to look at next. So I think for the past 2 or 3 years, my lab's been really focused on cardioception, where we've been trying to come up with better measures specifically in the cardiac domain. If you look at the interoception literature, a large proportion of the results over the previous 30 years are all in the cardiac domain. And that is more or less, as far as I can tell, simply because it's the easiest domain to study. You hook someone up to a pulse oximeter, you ask them to count their heartbeats for 30 seconds, boom, you get a score, you can correlate it with stuff. It doesn't require, an engineering degree to figure out. And that's what this task looks like, the so-called Chandri task, developed in the mid-1970s. It's basically exactly as I described. You attend to your heartbeats, you count them silently. There's some randomization, so you count for 25, 30, 35, 40-second intervals, and you make a confidence rating there so that you can calculate some correlation between the accuracy and the confidence score. And this task has been under a lot of fire recently. I'm going to cite this classic study. I had a nice chat with Hugo Critchley where he kind of said, I don't recommend emphasizing the study because it's a 90s study with about 7 people in it. So I think that's a fair enough point, and we would love to see a replication of this study. But you kind of have to lead with it. It's a classic study where they had people with pacemakers, and they ramped mechanically their heart rates by almost 100 BPM ground truth, and asked them to, before and after each measure, to rate how much they think their heart rate will change, if you're sitting or you're standing or lying down, and how much their heart rate actually did change. And basically what they found, you can see here in the counted heart rates versus the actual heart rates column, was virtually no change in the counted heart rates as the actual cardiac signal was changing, but really most of the variance in the subjective data, in the counting data, being related to their expectations about, when I'm lying down, I'll probably have a slow heart rate, or when I'm standing up, it might be a bit faster. And that's something that comes up again and again in the criticism of the heartbeat counting task, is this idea that it is probably just strongly biased by your prior expectations or just your subjective beliefs about what your heart rate is, if you have a 60, 70, 80 resting BPM. And I think that criticism makes a lot of sense in the context of how the task was originally proposed. if you go back to the 70s and 80s, the papers were arguing that this was a way to measure the kind of psychophysical sensitivity to single beat cardiac afferents. So the idea that this was really something that could allow you to detect sensitive signals from the cardiac system to the exclusion of things like beliefs and tactile sensation and, hearing your heartbeat pulse through your ears, like I do sometimes. And basically, there's been a lot of arguing over that and whether or not it does that. And what we're going to essentially argue in our approach is that if you really care about the cardiac, the ability to detect single beat cardiac afferents, meaning, without any other exteroceptive source of information, you should probably use something like a method of constant stimuli. You should probably not use something like a heartbeat discrimination task. The reason for that is very simple. The heartbeat discrimination task is one in which you hear a series of tones, and those tones are either offset to the rising wave of the systolic pressure wave, or they're offset to the diastolic period, let's say roughly 128 post-R wave versus a 384 milliseconds post-R wave. And you might wonder, why 128 milliseconds post-R wave? Well, it's true that does correspond to when the systolic pressure begins to rise, so you would assume there might be some increase in baroreceptor activity. But if you go back to the original papers, Whitehead, who developed the task, basically just assumed that 128 milliseconds would be when most people should feel the heartbeat. So this was treated as signal plus, and this was treated as signal minus, and then good old-fashioned signal detection theory could come in and solve everybody's problems. But basically, multiple experiments from Jasper Brenner and other colleagues have shown that if you use a method of constant stimuli to titrate the offset intervals for each signal plus and signal minus period, what you find somewhat unsurprisingly is that people vary massively in when they actually feel their heartbeats, meaning that my signal plus is very unlikely to be your signal plus, and the observed extremely low accuracy rates on this task are probably nothing more than presenting people with intervals they don't hear simultaneous to the heartbeat. So if you want to measure single beat intervals, I would run the Brenner task. It'll probably take you about an hour of testing. It requires it requires a good deal of sophistication in your temporal setup. You need to be able to deliver very precise temporal intervals. There are some open source methods coming to do that, using Arduinos and other things, but that is what you need to do. I would argue, however, that the ability to detect a single beat, it might tell you something about the sensitivity of your brain to the visceral system, in the same way that understanding if the visual system can detect a single photon is interesting for the basic neuroscience of vision. But I don't really think it's going to tell you that much about things like disorderly brain-body interaction in clinical populations. I think that our phenomenology of cardiac interoception is a lot more vague, a lot more transparent, or kind of not there. To put it another way, I think that most of the time when we sense our heartbeat, it's because we've just ran up a flight of stairs and systolic blood pressure is massive, so we can literally hear it or feel it somewhere. Or because we are afraid or we're anxious and we feel like our heart is racing independently of any kind of specific cardiac beat-to-beat feeling. So we wanted to try to build a task that wouldn't treat the heart rate as a pure confound or something that's not interesting for the study of interoception. We thought, rather than just asking someone, what do you believe your heart rate is, 0 to 120, which could be useful, we thought, what if you could actually have a psychometric method that would allow you to model the trial-by-trial variation in beliefs about the heart rate as you manipulate them using things like a blood pressure cuff or a pharmacological intervention. So to do that, we designed what we call the heart rate discrimination task. I am way over time, I'm sorry. I'm not over time yet, but I'm over time to finish. So you're going to miss the respiratory part of the talk at this rate, but that's okay. In the interoception condition, basically what happens is you have about 10 seconds per trial, 15 seconds with confidence. You listen silently to your heartbeat for about 5 seconds in a similar kind of condition to the heartbeat counting. You then immediately begin to hear auditory feedback, in this case tones, but it could be visual feedback, at a certain BPM. And you have to decide as quickly and accurately as possible if that feedback signal is faster or slower than your heart rate. And then you rate your confidence. In the background, we're recording the average BPM during this window, and we're using a Bayesian psychometric technique known as psi to estimate the minimum difference you can reliably discriminate between the feedback signal and your true heart rate. We call this the delta BPM because it's literally the difference between your true heart rate and the signaled heart rate. You can see here on the right how this works. So for technical reasons that are going to be in the paper, we started with an in, up, and down, where one staircase starts high. So this is a stimulus where you hear your own heart rate plus 40 BPM. Obviously, you really quickly know that's not my heart rate. I don't have a 120 BPM right now. Or you hear the negative 40 minus your own true BPM. And then over time, depending on your answer, that this is faster or slower, you can see that the staircase is narrowing in on a value that's closer and closer to zero difference. And then at some point, it's stabilizing around a threshold, which you can think of as the point of subjective equality between the perceptual exteroceptive tones and the internal representation of the heart rate. You can see that what the method does is after it finds the threshold around about here, it starts to test the extremities of the psychometric function, which is fit for this example subject here on the right-hand side. We also have a matched exteroceptive control condition where you either listen to exteroceptive tones and then again compare a feedback signal to say whether the feedback is faster or slower than the exteroceptive tones. And one of the things we really like about the task is, as you can see, both in the interoception and the exteroceptive condition, the performance is measured in the same units of delta BPM. So this allows you to kind of compare temporal biases more generally versus specifically interoceptive biases about the heart rate, like if you persistently believe that your heart is faster than normal. In this example, you can see that this subject has a threshold of about negative 20, meaning that they perceive or they believe their heart rate to be about 20 BPM less than average. And you can also kind of see here that for this person, the slope, which is a measure of the precision or the uncertainty of that decision process, is also much flatter for the interoception versus the exteroception. Now, this is just a single subject. When we look across 215 subjects that we collected this summer, if you can believe that, we see that this becomes a very robust finding. So while exteroception is generally unbiased in this sort of temporal comparison judgment, we can see that interoception shows a robust negative bias in the first sample we ran of about negative 7 BPM at a resting state. You can also see that the inter-individual variance around the interoceptive threshold is much, much higher. So there are people within the sample who are drastically over or underestimating, but on the average population level, there's a slight underestimation of heart rate. Similarly, for the slope, you can see that the interoceptive precision is substantially lower. So here, a higher slope number means a more flat psychometric function, which you can think of as having more uncertainty around the point of subjective equality. When we look at the metacognitive level, one of the things we really like about our task is it allows you to collect as many as 100 to 200 trials in just 20 or 30 minutes. And those trials are at or near threshold, meaning there's a lot of variance in the confidence to accuracy distributions. This is important when you fit computational models, such as the signal theoretic models of metacognitive insight, which essentially express a receiver operating characteristic for the probability to be highly confident when you make correct responses versus the probability to be highly confident when you make errors. The idea being here that if you have more area under this curve, your subjective rating is both more sensitive and specific to your ground truth accuracy. Now that's great, but in typical interoception tasks, you're fitting this type of model to data where you may have as few as 7 to 30 trials, you may have hit rates that are largely at chance, and that can cause a lot of problems for fitting these metacognition models. So we wanted to be able to also look at metacognition on this task. When we look at the confidence ratings, you can see here just in the raw data comparing exteroception to interoception that there is much better metacognition for exteroception. So you can see that the distribution here is much steeper, meaning that generally people know when they're making errors or not. Whereas here, there's still some dissociation. It's not that there's no metacognition for this trial, but there's a lot flatter, a lot less precise metacognition. And we actually fit in the paper a hierarchical Bayesian model of this M ratio effect, which essentially is the area under this curve standardized for the D prime for each subject, so standardized for the perceptual sensitivity for each subject. You can read this in signal theoretic units. So roughly what this means is we find that compared to a matched exteroceptive control condition, people lose about 20% of the available signal when they introspect on their interoceptive judgments, which we think makes sense given that the interoceptive judgments themselves are pretty imprecise. Finally, when we look at cross correlations between and across, within and across the modalities, here we were worried, maybe this task just measures like your general temporal bias, right? If you tend to perceive things a bit faster or slower than they really are. And if that were the case, we would expect a lot of correlation between, say, the thresholds and the slopes and other performance parameters between the intero and extero condition. So all this is an exploratory correlation matrix. Everything here is in the upper quadrant is corrected for multiple comparisons. And what you can see is that, largely speaking, there's really very few to no correlations between the interoception domain and the exteroception domain. There's a lot of correlations within each domain, but generally speaking, there's not an overwhelming amount of variance between the two conditions. There's a few things like the reaction time and the confidence that we can talk about later, but by and large, there's not a lot of overlap. But what about the face validity? So does the task actually relate to the heartbeat counting task? We also measured that in the participants at time 1. And this is probably really small now, but you can basically see that for the interoceptive condition, but not the exteroceptive condition, there's a highly significant, but modest correlation between the interoceptive accuracy scores on the heartbeat counting task on the x-axis and the HRD thresholds on the y-axis. So essentially, the more you undercount the more you underestimate your heart rate on the HRD, the more the lower accuracy you have on the heartbeat counting. And that corresponds nicely to previous reports that heartbeat counting scores are mostly driven by undercounting of the heart rate.
Micah, just one note. We didn't see where you were pointing. I just realized that you've been pointing. Sorry.
Oh, you don't see the mouse. Okay.
I guess it was in another screen, but it has been clear so far with your explanation. Just now, I didn't realize which one was what. Sorry.
I'm definitely gonna have to skip the respiration, but maybe that's okay. There's like 160 people here. It's making me sweat a little bit. So what does that mean? Well, okay, it means that something like I can't do the square root of 0.29 in my head, but there's some shared variance between our task and the heartbeat counting. We now basically think that the heartbeat counting is a kind of noisy measure of roughly the same process, but our task is separating that heart rate estimation effect into the bias, the accuracy, and the sensitivity, and we think it's ultimately going to give a better grasp on how those heart rate beliefs change over time. Now, what about test-retest reliability? So we invited the subjects back at time 2. Only about 100 or so came back for the second session due to many logistical issues. But what we see for the thresholds is overall, we'll call it a good to moderate reliability. So this is test-retest at around 2 to 3 months following the first session. There's also some caveats, or rather there's some kind of limitations in that we don't actually keep everything the same between session 1 and session 2. There are some things under the hood that we changed. Oh, someone says they're here for the respiration part. Okay, I won't go all into this. Basically, we see, I think, surprisingly good reliability for essentially how fast someone thinks their heart is beating at rest. This is months apart, and you can still predict a good deal of the variance from the session 1 to the session 2. And for reasons we'll go to in the paper, which should be pre-printed this week, we think that this is a lower bound. We actually are pretty sure we can get even better on the threshold here. I don't show you the slope. The slope does not have great reliability right now, but we can talk about why that is later. I'm going to kind of skip this part because it's a bit redundant. It's just showing some of the test-retest effects. I think the biggest takeaway is that threshold is super stable across modalities, so there's no session by modality interaction. Exteroception is generally unbiased, interoception is generally biased. People go down a lot in terms of their uncertainty, so they become more precise in both modalities from session 1 to session 2. They get more sensitive in terms of D prime, but the greatest improvement is seen for interoception and also for confidence, which could just suggest that the low precision of interoceptive belief makes it more amenable to learning effects It could also have a note of promise for clinical intervention studies. Maybe if you run our task with true or false feedback, you could remediate some of that bias. So that's the summary of the Cardioception. We think that the heart rate discrimination task does a pretty good job of measuring the accuracy and the precision of cardiac beliefs. So we like that we think it measures the thing we say it does. It delivers those measures in sensible units, which can be directly compared between exteroceptive and interoceptive controls in the same units. And it gives reasonably good reliability. We have a lot more work to do. There's going to be, in the future, we're going to be doing a lot of contextual manipulations of heart rate and arousal, pairing that with computational modeling. We think that this task is really nicely amenable to modeling the underlying decision process that drives changes in the heart rate belief. And this is going to be a big focus of research for us in the future. If I can be allowed 3 minutes, I'll race through the respiratory part. I'm really sorry if anybody needs to go or you're waiting and I'm not on time. I try to always be on time.
Go for it. People are interested, which is cool.
So that was the cardiac domain. What about respiratory interoception? So the short story of explicit respiratory interoception is it's like a little bit neglected. So there are a few papers here and there that measure our ability to detect sensations in our lungs and our throat and our airway, but they have nowhere near the attention given to cardiac. Currently, the best task on the market, we think, is developed by our collaborator, Olivia Harrison, which is a filter detection task. The nuts and bolts of it is you breathe through a circuit, and the experimenter manually adds or subtracts physical filters that increase the resistance at the end of the circuit, and it gives a coarse measure of the respiratory threshold in terms of the minimum number of filters you can reliably discriminate. This is something we ran in Cambridge in a pilot study comparing obese versus lean subjects to see if they have any differences in respiratory interoception. But the message about this task is that it's really painful to run, basically. It takes about an hour to get 60 trials. The measure of threshold is very, very coarse and is not necessarily controlled for things like the inspiratory pressure at the mouth. And it can actually be quite unpleasant for psychiatric patients because it involves a lot of heavy breathing. So we wanted to develop a new approach to measuring respiratory interoception. We call this the respiratory resistance sensitivity task, developed by really excellent work from my postdoc, Nia Nikolova, where what we did is we took the same concept. We actually cannibalized all the same parts from Olivia's task, things like you can see here in this picture, little sanitary one-time use only mouthpieces, a T-valve to control dead space, some standard respiratory tubing. All the funny neon bits are 3D printed, basically. So the funny little green box, this is what does the magic, so to speak. What happens here is this little pin is controlled by a 3D printer step motor. And that step motor is controlled by an Arduino so that it can compress a flexible tube that you see here in very, very small steps, fractions of millimeters with this current setup. And as that tube gets closer and closer, there's a nonlinear relationship between the pressure that you exert on the circuit and the inspiratory resistance that you feel. So unlike the previous task, this allows us to use a psychometric approach, again using the same PSI procedure or Quest procedure if you prefer, that we talked about in the cardiac section, to allow the algorithm to control the step sizes and the signal presented to the subject to really quickly hone in on their respiratory thresholds. So in the task, you basically see a visual feedback. It tells you to inhale as long as the circle is expanding. You then exhale, you inhale again, and you say which of the two breaths had a load on it. One is a signal zero trial where the pin is not touching, and another is a signal present trial. You can see here, for example, for this subject, that in just about 5 trials, the Bayesian staircase procedure has found a reliable threshold. So this is something we're actually piloting now. We're up to about 20 subjects. I'm going to kind of gloss it. We also have people rate the subjective unpleasantness of the task over about 45 minutes of testing while we're validating the task. And you can see in this example subject that generally speaking, we have pretty good ratings. So this is a subject that kind of started low and after about an hour of testing was getting to about, say, 35 out of 100 from pleasant to unpleasant. Oh, no, I think it's totally unpleasant to, it's not at all unpleasant to totally unpleasant, sorry. So the great thing about this is it allows you to measure your respiratory thresholds, we think in as little as 15 to 20 minutes. If you can go longer, say to 80 trials, about 30 minutes, you can actually measure the psychometric slope again and get a measure of respiratory precision. We're doing a little experiment now where we're running a counterbalanced order. Subjects take 30 to 35 minutes of the psi staircase and then 15 to 20 minutes of the quest staircase, so we can compare the reliability. You can see, for example, in this threshold, in this subject, it's done a very nice job in both cases of converging to the same point. Here you can see over our current, I think we're at 17 subjects, you can see all the different staircases and the different stimuli they present and the psychometric functions we fit to each respiratory performance for each participant. And something I'm really happy about, this is really preliminary because we're still collecting the pilot data, so ignore the p-values. This is just spit out by ggStats plot. But I think the good thing is that at least within a single 1 hour session, where you counterbalance the 30 minutes of testing both staircases, you get a very high reliability in that respiratory threshold. You can also see it here that there's very little within individual variation between the threshold estimates. And the overall average ratings are pretty stable as well. They're not really getting more unpleasant as the task goes on, and the average is somewhere around, a quarter of the total scale, so like maybe mildly unpleasant. So there should be a summary about this. We're really excited about this task. Both of the tasks you saw here will be released as open source Python packages. So we've already released systole, which is what the heart rate discrimination task is built on. This is an open source package for interfacing with pulse oximetry data and soon ECG data, so that you can start synchronizing your stimuli to the heart rate however you want. And pretty soon, I think about 2 weeks from now, you'll be able to type pip install Cardioception, and that will give you a command line interface so that you can actually test the heartbeat counting or the heart rate discrimination test in your own labs. The same will be true for the respiratory task. For that one, we will actually be releasing the AutoCAD files so that you can 3D print your own apparatus. All of these measures take about 300 to 400 euros in total per unit. We're actually running 4 parallel respiratory apparatus in Aarhus. I'm rushing, I'm sorry. And that's basically it for the talk. So our group has a lot of work ahead of us. We have about 200 people tested on these tasks with fMRI and other neuroimaging biomarkers. So in the coming year, you're going to see correlations between interoceptive psychometric functions and brain-body biomarkers. We're going to be looking at psychiatric symptoms, predicting depression and anxiety. I don't know if we'll find anything there. We're really excited to start doing clinical and pharmacological studies, as well as preclinical and translational studies. We have several applications and collaborations developing to do things like interoception in pigs, in rodents, and other things where you can actually get in there and causally manipulate. And we think really that the future of this research is using tasks like these, where you can model the trial-by-trial decision process associated with an interoceptive belief or an interoceptive percept, and relate that to causal manipulations in arousal, in prior beliefs, do things like emotion induction, do things like give pharmacological substrates that alter the brain-body communication. And starting next year, we'll be beginning our ERC starting grant called Cannabodies, which is going to be taking all of this research approach you see here and systematically in randomized control trials looking at how THC and CBD modulate brain-body communication and interoception. So we're really excited, and we also have a lot of postdoc and PhD positions opening soon, so get in touch if this is exciting for you. And that's all. Thank you very much. I'm sorry I went over a lot, 15 minutes.
You didn't. No. You went over 10 minutes, and it were 10 minutes of very interesting information, so I appreciate that. I think the community also appreciates, so thank you. It was really interesting. I think we can go to the questions now. So we have already some. I'm going to go through the most voted ones. It's easier for me. So the first one, which I also had, it's how would this relate to meditative states and mindfulness? And I guess this is coming from the heart rate perspective, right? Because I really do feel like a lot of these the goal of these, at least in the beginning, as far as I've experienced, is to control your heart rate. That's really a lot of the focus of these meditative states and mindfulness. I will add, do you think that actually controlling your heart rate, for example. Not sure if this is where the person was going, but if it actually will allow you to react differently to situations that might be somehow encoded. And that's why people are really trying to do that because of that.
It's a good question. Sorry, I laugh because I started my research career.
I can imagine this is
I actually, my PhD was on mindfulness, and I kind of ran away from that field, but stayed with the interest in embodiment. And I think it's particularly as we get into respiratory interoception, it's inevitable that I can't get away from the mindfulness, but that's okay. It's an interesting research area. I think and all apologies to any mindfulness researchers in the field, my impression when I left that field, which was maybe 7 years ago, was there was a lot of really confident beliefs about how mindfulness is extremely important for changing your metacognitive awareness of interoception, but really little evidence. So there was sort of one famous study from Sabib Khalsa and other colleagues, I think working with Damasio, where they used a heartbeat counting task, and they showed that really advanced Tibetan monks were really confident in their cardiac interoception, but no more accurate. So they were kind of, in a practice where they were being told that they're contacting their heart and they're doing heart practice, Dzogchen meditation. They had really strong beliefs that they were better at a cardiac task, but we didn't see that. So what I would really like to see would be, systematic randomized control trials looking at mindfulness and meditation practices in cardiac and respiratory domains to first of all get that evidence and see that there is a strong and specific treatment effect, compared to something like a yoga control or a even simple relaxation response control. Now, all that being said, I did used to practice meditation. I think in a past college life, I was definitely a Buddhist at one point. And I think that, what I used to say when I did mindfulness research is that there was one mechanism of action I could be convinced. It probably had to do a lot with awareness of the breath, and things like Vipassana meditation, many forms of mindfulness and contemplative practice. Being aware of and centering on the breath is such a core part of the practice. And in my, current life, I think that's one of the skills I retained from that period is, just pausing and noticing what the breath is doing. And to answer the heart rate question, we often forget that the heart and the breath are very closely linked. There's something called the respiratory sinus arrhythmia, which basically means that if you hold your breath, your heart rate skyrockets. So I think that there's a lot of clinical potential in learning to notice how you're breathing, and if you're, overexerting or hyperventilating, and learning to slow down and regulate the heart rate that way.
I totally agree. Really cool. Okay. Another question that we have is how related are interoceptive measures or abilities between modalities, meaning cardiac, gut, respiratory?
That's a good question as well. So in our task, I can maybe zoom back here. Sorry, there's a dog barking. In our task, not very, but that's maybe by design. So in our task, there are basically very, very few correlations between for example, here you can see I think this figure is wrong because this actually doesn't survive the FDR correction. But you can see the plot here of exteroception threshold versus interoception threshold. There's if you squint through the shotgun blast, maybe there's some relationship. The only thing we really see a strong multi-domain correlation in the cardiac domain is confidence. So being really confident in one task is very strongly related to being really confident in the other, even though the D prime, meaning the objective sensitivity, is not related. So to put that another way, it seems like if you're just a generally overconfident person, that relates to both tasks, but it doesn't mean that you're any better at the task. And that's probably some of the metacognition effect we're seeing, where people don't seem to be updating that confidence belief as well. But it's a little complex because basically this type of confidence is almost like a trait, right? If you tend to be overconfident, you're going to be having a higher confidence on every task. But of course, when we look at these tasks specifically, on average, people are much less confident for the intero versus extero condition. Now, aside from that, it's hard to say. We lack good gastric tasks. It's very hard to do gastric interoception without going fully invasive. There's one study from Sarah Garfinkel that found, I believe, some correlation between respiratory and cardiac interoception. I think the best answer is right now, in considering the criticisms of the measures, the lack of large sample studies, I think we don't know. I think it would be a great project for something like a mini labs registered report to, get 20 labs, measure interoception in 3 domains, get some emotion measures in there as well, and see what the general reproducibility of those correlations is.
Yeah, cool. I see. Okay, so now we have another question, which is when we ask based on past 24 hours, I guess this is related to one of the tasks, can underlying health wellness condition for the day strongly bias the rating? Or is that intentional?
Sorry, one more time. I got distracted by changing.
Okay. So, no worries. So, Akshay Ravindran, sorry if I forgot to mention people's names so far. He asked when we ask based on past 24 hours, I think it's how people felt during the past 24 hours regarding their general state, right? Can underlying health wellness condition for the day strongly bias the rating? Or is that intentional?
Yeah. No, I mean, it's a subjective rating. So, in that study, they were getting an email at kind of regular intervals asking them to do, introspect on the past 24 hours. And for example, we see also some of those correlations I showed with mental distress. It also correlates with physical distress. And I think there was a really large paper out recently showing that in adolescence, the single greatest predictor of a reduction in mental health from the COVID-19 crisis basically has to do with the disruption of a physical daily routine. So, I think it's really important. I'll scroll to those data really quickly, but it's really, really important to underline that, like I said, we don't know, are these effects something you might call a functional effect or a hypochondriasis? Are these, is this an adaptive response to, monitoring interoceptive signals? I know that myself, particularly in March when we did this study, every time I went outside and went to the grocery store for like 3 hours afterwards when I got home, I felt like I was constantly checking, do I have a fever? Is my throat weird? Because, that's what a pandemic does, is it churns visceral signals into threat signals. But there's no causation in the study. It's purely correlational. And we don't know whether these subjective feelings, these self-report measures are indexing, that people are actually anxious. So they're having more gastric events. It's just, again, speaking to that, we can't dissociate the subjective belief from the ground truth in that type of measure. But we still think the MIES is really useful as a better measure of subjective experience, because we've tried to cut out all the fat and focus in on just the kind of phenomenology of experiencing the body. And we've tried to do that in a way where you could use like a smartphone study. We actually applied for money and didn't get money for that. We wanted to do like a large scale international study following up on post-COVID patients to see if those elevated interoceptive experiences might actually predict later mental health outcomes. But it's still something we're looking at.
That's interesting. I guess we've never been in a situation that the whole world is kind of, we've been in not this type of situation. It's interesting because it's the same pressure being applied to everyone, and still people tend to react differently depending on it's really cool.
I mean, our feeling has been like, we sometimes joke that interoception research feels a bit on the fringe, but it doesn't feel very fringe anymore. It feels like we're all undergoing an interoceptive crisis, basically.
That's true. That's a good way to put it.
I didn't say it, but something we're actually doing with the respiratory task. We have a collaboration in Denmark, this one. So we're actually measuring respiratory interoception in a large sample study of post-COVID patients with the so-called long COVID. So we're trying to use this model to actually try to understand, is the 6-month post-infection dyspnea, breathlessness sensations you see in long COVID patients, is that something that's more at the level of subjective belief or objective physiological change or the interaction there? So it again drives home for us, that it can be important to study this stuff.
Yeah, that's really cool. Yes, didn't think of that way. That's interesting. Okay, so a final question from Leah Benelis. So she asks, how will you test interoception without requiring a response, for example, in animals?
It's a great question.
I think it's a great question to end.
And the whole time before my talk, I was thinking, these are animal neuroscientists. I should really be thinking about this. What a good question. Yeah, we joke about that a lot. Like, could we train a pig to, sniff one way or another? So, I mean, you can train a mouse to do a random dot motion task, right? So you can actually have, like in Amir Locke's very excellent work, you can have a mouse, running on a wheel, and it can sniff one way or another for if the motion is left or right. So I don't know. We've said that maybe it could be possible to use some kind of feedback, like the mouse gets more reward if it sniffs when the signal is synchronized to the heart or not, or maybe a monkey. I don't know about measuring explicit interoception in that way. What we're looking at in animal models right now primarily, and we see in the literature actually a huge number of papers coming out, is doing stuff that you can't really do in a human model. So things like invasive VNS, directly electrically stimulating different visceral parts of the vagal nerve, doing things like manipulation of satiety, water deprivation. So there was a beautiful paper by Livna et al. In Neuron last year where they looked at how rodents learn about basically operant conditioning under threat when they're in satiety or non-satiety states. I think there's a lot of room we can do with these invasive and causal techniques in systems neuro to start to build that causal picture from the body up. I would love to hear some ideas of how we can measure explicit interoception in animals, because I actually think it's not impossible. I don't have the secret sauce yet, but I would like to give it.
Well, I have a dog, and he kind of tells us when he's happy or not. So I'm joking. we might be able to get there. Okay, this was a really interesting talk. Thank you for also making the effort to cover everything. I think our crowd is really happy that you went to the respiration part. So cool. So I would like to thank you for being here today. We will now move to the Meet the Speaker session, like in a couple of minutes, just to have a break. So the ones that got the email, please follow the Zoom link. If you still want to join, just send us an email, and I'll make sure I'll send you the Zoom link now. So thank you. We will see you next week with Dr. Anna Molovsky, and she will be telling us about microglia, memories, and the extracellular space. So yeah, thanks, and have a great week, everyone. Thank you, Micah, once again.
Thank you so much for having me, and